And no one wants us to start over again. Anyway, well, hi everyone, and as we said, thank you so much for joining us today for what will be the first edition of our UCLA CERM Thermo Fisher Scientific Stem Cell Basics course. Again, my name's Tom Forbes, and I'm a staff scientist at Thermo, where I support our stem cells and neurobiology portfolio. So what we're going to do today is kick us off with our introductory lecture, which I'll introduce shortly. But before we dive into that, I'm just going to spend 5 to 10 minutes or so introducing the course itself and talk about just a couple of our objectives in leading the course. And as well, just some of the folks that will be sharing their vast knowledge of stem cells with you. Really, our overarching goal for the course is to provide you with a foundational training on basic stem cell techniques. This is, of course, as you see here on the slide, going to include topics like stem cell culture, reprogramming, gene editing, differentiation. And I'm sure many of you are quite familiar with, or at least partly familiar with some of these concepts, and it's totally okay if you're not as well. But all of this is going to be led, which I think is really, really cool. And it's always a nice collaborative approach here by the faculty and staff from both UCLA and Thermo Fisher Scientific. As Catherine alluded to right at the beginning, the course represents a component of the UCLA California Institute for Regenerative Medicine, or CIRM, as we affectionately call it, a shared resource activity. CIRM, which I'm sure many of you have heard of, is quite a massive state agency that's established by the people of California to help accelerate things like stem cell, regenerative medicine, and gene therapy research. We get a good amount of funding for CIRM, which then helps support things like development of critical research innovations, and also, which is why we're here today, invest in training the next generation of scientists, which is all of you guys to help us lead the way moving forward in advancing things like regenerative medicine. So as you see here on the slide, we're going to be talking about things like best practices for aseptic stem cell culture, which is obviously a massive part of successful research. Human somatic cells, so things like blood and skin cells, for example, being converted or reprogrammed to stem cells, the so-called iPSCs, or induced pluripotent stem cells. We're also going to talk about some of the basics of general stem cell culture techniques, think expansion, maintenance, passaging of stem cells, things like that, as well as some handy tips and tricks from folks like us along the way. We're also going to be talking about differentiation. That better? Sorry about that, everyone. It happens, though. There's always glitches on the first day, which is totally fine. As I said, we're also going to dive a little bit into gene editing. We'll talk about as well some of the clinical applications of stem cells in regenerative medicine. And one thing that Omar and I will drive home throughout this course here is your community, right? We have collectively managed to gather a number of subject matter experts here in the field, all of which, I kid you not, have such a wide array of experiences, connections in the field, and obviously vast knowledge underlying all of that. So please, 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 in taking this course, leverage that. Use this course as a unique opportunity, which it very much is, to expand and enhance your professional network within this stem cell community. It's important. And it's important to take advantage of that as well. Now, as far as specific content for each of the 7 lectures here, we're going to have 7 discussions, as I said, or lectures spread out over the next couple of months. We're going to meet Mondays at 9 a.m. PST, unless you hear otherwise. The lectures will be about an hour to an hour and a half, I'd say, for each session, although we have allowed for a bit more time at the end of each should we need it. Each of the talks, as you see here, is going to be centered around specific topics, things like introductory stem cell culture, reprogramming, feeder-free culture, gene editing, differentiation. And then again, from these different discussions, what we're hoping to do is provide you with a fairly comprehensive foundation in stem cell culture, in which we'll also highlight some relevant case studies and literature, and kind of give a nice historical perspective as well to touch on some of the past, present, and future of what's become a very, very much innovative and ever-evolving field. Again, we're going to share with you quite a lot of science-based tools that we're lucky to have at our disposal nowadays. Again, just to drive it home, especially institutions like UCLA, paired with companies like Thermo Fisher, where we are really fortunate to be surrounded by a lot of cutting-edge resources and talented people. Anyone have an idea what these cells are on the right? I know most of you are muted, so you might not be able to say, but perhaps you can pop some ideas into the chat. This is an image that I stole from Omar. These are mouse embryos that we collected at the blastocyst stage here. So they're about 3.5 days post-fertilization. You can see that each of them is a little bit different looking, right, from one another. But developmentally, they're all roughly at about the same stage. And we're going to get more into that kind of thing and those kind of examples shortly. Again, I've put together a list of our course instructors here for their associated contact info. Please feel free to reach out if there's anything that you need, or if you have any specific questions as well. We're all happy to be here and happy to help lead this course. And then lastly, before I dive into today's discussion, I did want to touch on course materials. As I alluded to, we're going to be sharing the presentation slides after each lecture. The lectures are recorded, as you know, as well, which is handy. We'll also provide you with things like associated protocols, different case studies, and links to quite a lot of free and completely useful resources that pertain specifically to each lecture. And I'm sure some of the folks from UCLA will add to these resources as well. But the long and the short of it then really is that we'll be sharing a lot of useful content to support you during and after this course. And again, much of the work that we're doing in this course is very, very much integrated and shared amongst us. So feel free to leverage your instructors as a resource as well. Again, whether that's scientific questions, experiences that you'd like to share, or even offline career development as advice. We're excited to lead this course, and we're really glad that you signed up. So please don't be shy.
I will just reiterate that, Tom.
Yeah,
Please
please.
take the opportunity to engage as much as you can, right? There's, no such thing as a stupid question, really, honestly. Anything that is not clear that you want a little bit more color on or that you're interested in, or as Tom mentioned, if you're wanting to dive into one of the resources a little bit better, you have quite the unique opportunity here with senior and very distinguished members of the UCLA faculty, to support, but as well, you also have Tom and myself to bounce ideas off of, to really get some advice, whether that's career related, project related. We're definitely happy to help.
Yeah. And a really nice balance between what's currently happening in academia and what's currently happening in biotech too. And I think that merger is really special. So this is a really unique and good opportunity for you guys. Anything else, Omar, before we kick us off?
You're all good? We're ready to
Okay.
roll.
All right. Well, we're going to kick us off then, as we've been saying, with an introductory lecture. With regards to the specific content that we're going to cover today, we'll talk a little bit about biosafety in the cell culture setting. Admittedly, this isn't always everyone's favorite topic, but we're going to spend 5 minutes or so on this, and it's really important information to keep in the back of your mind. We're then going to segue into some basics surrounding stem cell culture before transitioning over to kind of a high-level overview of some of the science and science-based tools underlying the cell culture workflow, especially that Omar and I support in our roles at Thermo Fisher. And then lastly, I thought it'd be interesting to round out the discussion with a case study. For today's case study, I've chose one that pertains to Parkinson's disease in 2D stem cell culture, and one that I think nicely incorporates a lot of the tools that we'll be talking about today. Sound good? All right. So again, as good a place as any, I think, to start with a cell culture course is biosafety, which I'm sure you've heard a lot of this information before, but honestly, it really, really is worth reinforcing. Even a small deviation away from biosafety in the lab can quite severely impact your health, right? Not to mention the consequences of the lack of things like proper sterile technique can have on the outcome of your cell culture experiments. And so really a cell culture laboratory has a number of hazards associated with it, with the handling and manipulation of different cells and tissues, things like toxic, corrosive, or even mutagenic solvents and reagents, right, that are all around you in a lab setting. And really, so the fundamental objective of any biosafety program is to help reduce, or even, we hope, eliminate exposure of researchers to those potentially harmful biological agents, and then reinforce adherence to standard microbiological practices and techniques, right? The regulations and recommendations for biosafety in the US are contained in quite a big publication called Biosafety in Microbiological and Biomedical Laboratories, right? And basically, the document outlines quite a lot of things, but it does define 4 ascending levels of containment, referred to as biosafety levels 1 through 4. And these basically describe different microbiological practices, safety equipment, and facility safeguards to help us reduce risk that's associated with handling, say, a particular agent. I've highlighted in blue here, the second one down, right? Most culture labs that we'll be working with should be at least a BSL-2, but obviously the exact requirements depend on the cell line that you're using, the type of work that you're conducting, etc. But in the interest of time, we're not going to go through each of these, but again, I will call out BSL-2, which I think is appropriate for moderate risk agents that are known to cause human disease, typically through mechanisms like ingestion or through mucous membrane exposure. Again, I realize this time around we're not going to be going into the labs together, but I do think it's really, really important to touch on just a couple of practices that help us create barriers between you and these potential hazardous agents that you're going to be encountering, right? As well as protecting your cell culture experiments from contamination by you, which, as careful as you might be at all times, this can happen, right? So PPE, or personal protective equipment, is really the first line of defense. It's what forms an immediate barrier between you and any potentially hazardous agents, right? This will include things like gloves, lab coats, gowns, safety glasses, right, which you should always wear in the lab. It's common sense, right? This also can include things like shoe covers, respirators, face shields, right, that we can wear for higher exposure experiments. PPE is also used in combination with biosafety cabinets and other devices that are in the lab, right, to help contain those potentially harmful agents. And so really, these are all general guidelines for safe laboratory practice. Always wear your PPE, change that PPE if and when it becomes contaminated, and please report any incidents, right? It doesn't matter if they're big or small. You have to report them to the appropriate personnel at your respective institution. Now, as I kind of alluded to before, the specific requirements of a cell culture lab always vary depending on the type of the research that is being conducted. But all cell culture laboratories have a common requirement for being free from pathogenic microorganisms, right? And they typically then share some of the same basic equipment that's essential for culturing cells. This will include things like cell culture hoods, right? These provide you with an aseptic work area. They help contain infectious splashes or aerosols that are generated by a lot of the procedures that we do. They also protect your workspace by maintaining a constant flow of filtered air. That's super important, right? On the right here, we also have a fellow standing in an incubator, which helps us maintain things like temperature, CO2 levels, humidity levels, basically provide you with an appropriate environment for cell growth, therefore mimic conditions in the body where those cells perhaps originated. Now, other pieces of commonly seen lab equipment include things like microscopes for imaging and counting cells, hemocytometers, right, which we use for counting cells. We also have refrigerators, water baths, centrifuges, media aspirators, different forms of cryostorage, sharps containers and biohazard containers, right, which are always red and they have to be labeled appropriately. And again, I'm sure a lot of you have a good amount of experience by now in a culture setting, but one day you might be running your own lab altogether. And so I think this is just a nice quick point of reference and a gentle reminder. And then lastly here on this, sterile technique is really, really important. It's such a massive component of stem cell culture. Obviously, it has two primary goals, right? We want to protect your cells for contamination by you. And then on a larger scale, right, protect you, the researcher, from exposure to those known and hidden lab risks. And so this slide really is just a gentle reminder from us to always work thoughtfully, right? Stay organized in the hood, maintain a clean to dirty flow, as you see here in this cartoon down here at the bottom. You should always routinely decontaminate your work surface and materials before and after each use. Clean up immediately after if you spill anything. Make sure you have as well a regular cleaning schedule for your lab equipment, even when it's not known to be contaminated. And another thing to drive home here that I think sometimes is lost in the fray and lost in the shuffle, we all get very busy, but remember, good lab etiquette, right? There's a lot of real estate in the lab. There's therefore a lot of different shared spaces. And so it's always super important to be considerate of others and not ruin anyone else's work by being careless, right? I think a lot of this is common sense for most people, but we want to help reduce the risk of exposure to potential hazards here because this will really help ensure that you're safe, right? And that your cell culture experiments will be successful.
Hey,
Now,
Tom.
switching gears here just a little bit to, I want to dive into a
Hey,
little bit
Tom.
of, yeah, go for it, Omar, please.
Sorry if just on the last slide, I just wanted to
Yeah,
give just a little bit more color here. Two very specific examples where you could kind of see those sterile techniques practices taking place and sort of really benefiting you in any cell culture setting, let alone stem cell culture, right? So you heard Tom talk about the clean to dirty kind of spatial distribution inside your hood. Also, it'll be helpful to think about the depth of the hood as well, right? So a couple of slides before, Tom showed the airflow circulation and primarily those BSC units are there to make sure that the airflow isn't introducing any pathogens onto your cells, right? Especially because we open our cultures inside of the hoods That being said, working as far into the hood as possible is recommended, right? So instead of working super close to the sash to try to place your samples or making sure that you're working a little bit further in. That being said, making sure not to block the entry points that those airflow ducts are pointing towards, right? So you see that there's ducts in the back of the all the way in the back of the hood. A lot of folks end up putting a lot of tips in there or a bunch of clutter that they might need in the hood, but then they might restrict that airflow. So just be aware of that it's good, as Tom mentioned, to make sure that your station is clean, that it's organized. And as trivial as that sounds, you get into the habit of knowing where things are this way, of making sure that you're utilizing all the space that you need optimally, but also all at the same time making sure that you're not introducing any pathogens to your culture while you're handling the cells as well.
Thanks, Omar. All right, we'll dive into a little bit of the science underlying cell culture basics here. Again, I know some of this content is going to be a review for some of you folks at this point in your particular program, but it's always important to touch on the fundamentals because this is kind of our building blocks that'll help us understand some of the concepts that we're going to be reviewing together. And so, what exactly are cells? Just kind of on the whole, right? These are the smallest units of life. That can be a whole organism, right? Like bacteria, yeast, parasites, or even a larger integrated part of an entity, right? Like a particular tissue, an organ, or even an organ system, right? But cells are the basic structural and functional unit of every living self-replicating organism. And there's two main classes of cells, right? We've got eukaryotes and prokaryotes. Prokaryotes include things like bacteria, archaea, cyanobacteria, those really cool blue-green algae, right? While eukaryotes include things like animals, plants, fungi, insects, and humans, right? We're composed entirely as humans of eukaryotic cells. Most scientists believe that eukaryotes evolved from prokaryotes about 2.7 billion years ago, with the primary distinction now being that eukaryotic cells have a membrane-bound nucleus and prokaryotic cells do not. The nucleus, as I'm sure you know, right, is where eukaryotes store their genetic information. Now, they're similar, but they're not the same, right? There's differences between these two main classes of cells, right? But they do share some common features, right? They have DNA, right? DNA is the genetic coding that determines all the characteristics of living things. And the entire DNA in a cell can be found in individual pieces called chromosomes. So both cell types have chromosomes. Eukaryotic cells have many chromosomes, which undergo, right, meiosis and mitosis, just to throw a few words around that I'm sure you would be a blast from the past and you remember from before during cell division, while most prokaryotic cells consist of just one circular chromosome. Both have a plasma membrane, right, which separates it from the surrounding environment and helps act as a selective barrier for incoming and outgoing materials. Anyone remember what the cell membrane is made of around the edges here? Phospholipids, right? And phospholipids have a net negative charge. We also have the cytoplasm in the middle here, and that's that jelly-like fluid within a cell. It's composed primarily of water, salts, and proteins, right? Speaking of proteins, anyone remember the name of the organelles that make proteins? Ribosomes, right? And the proteins are everywhere in cells, including embedded in the outer membranes here, right? They can also be secreted to affect other cells through different factors and cell signaling. What about nutrients and energy, right? How do cells get nutrients and energy that they need to survive? Well, cells from tissues are normally supplied with nutrients by blood, but in cell culture, right, we use different types of media to help us replace things that are normally supplied by, say, the bloodstream and that are needed for things like cellular metabolism. So remember, when we're talking about in vitro cell culture, right, that method of studying the behavior of animal cells, we want to mimic the setting of a living organism and then control for as many variations as we can to help recapitulate that cell's natural environment. And so that means, with regards to cell culture, that at the most basic level, we give this to cells to provide it with nutrients and offer them a stable environment in which to grow. In doing so, when we're making media and using the right appropriate media, we have to account for things like salt balance, sugar levels, essential vitamins, amino acids, pH, temperature, you name it, right? Modern media are quite complex, right? And they often serve a specific purpose or a specific cell type or type of culture. And we'll unpack some of that in a later lecture in this course. There's also the importance of different attachment proteins and different growth factors, right? Depending on the cell type that you're using, right? So take home here is that media choice and all those different factors help us control the cellular environment, and they're really, really important considerations. But what about stem cells, right? That's why we're all here. This is the important part of this course, right? What exactly are pluripotent stem cells, or PSCs, as we affectionately call them, right? These are quite remarkable cells that originate from, I'll put a blue thing on here, sorry about that. These are remarkable cells that originate from early stage embryos, and they really have at their core two defining characteristics. The first of which is the ability to self-renew indefinitely, right? And they also have the capability to differentiate into varied cell types from the three so-called germ layers, as you see here, right? Endoderm, mesoderm, and ectoderm. What that means is that they can give rise to just about any cell type in the body. How on earth does that happen, right? Well, as you see in this kind of nice schematic here that Omar put together, shortly after fertilization, what happens is the zygote here starts to divide. And as you see here, quite quickly, these 2 cells become 4, 4 cells become 8, and so on and so forth, until you get to this morula stage at the top here. This is where we start to really see those physical changes. And this cluster of rapidly evolving cells really starts to self-organize, right? Migrating, compartmentalizing, and almost in some ways beginning to adopt their actual fate. And it's here that we reach what's called the blastocyst stage. The word blastocyst refers to the stage that the human embryo reaches about 5 to 6 days after fertilization. An early blastocyst is composed of 2 different cell types. We've got an outer layer of cells called the trophectoderm. The trophectoderm gives rise to the extra embryonic tissues, so think like the placenta, the amniotic sac, as well as a group of cells located on the inside, which, being on the inside, they're appropriately named the inner cell mass, or the ICM, right? And those ICM cells are the ones that give rise to the embryo proper, or the fetus, as a pregnancy progresses. And so pluripotent embryonic stem cells originate from these inner cells within a blastocyst. Again, these stem cells can become just about any tissue in the body, excluding the placenta. And so think of the inner cell mass as what gives rise to the 3 germ layers, right? Our endoderm, mesoderm, and ectoderm. And so that's where the name stem cells come from. Those embryonic cells of the inner cell mass, which by definition are self-replicating, meaning again that they have the capacity to divide and could potentially give rise to themselves again, as you see here in the middle in this box, right? Call me biased if you will, but I think especially over the last 6 decades or so, stem cell biology has really, really become one of the most exciting, informative, and I'd say rapidly changing and possibly clinically transformative areas of scientific investigation. And I think I'm speaking to the right crowd here when I say that regenerative medicine especially really does have such great potential of providing things like curative approaches for a whole bunch of different disorders that originate at the stem cell level. And so, we often talk about this a lot at work, but I think the true power of PSCs lies in their regenerative medicine potential, right? Which has opened a lot of different applications. In basic biology, we know that PSCs can help us understand things like early human development, cell differentiation processes. We use stem cells as well for disease modeling, right? Because they allow us to create accurate depictions of human diseases. This, of course, enables better understanding of a lot of different disorders and treatment for, and often the screening of new pharmaceuticals. We use stem cells as well in cell therapy nowadays. They have a lot of promise to help us generate healthy cells to help replace damaged tissues, right? And again, even potentially offering cures for various debilitating diseases. And again, we'll unpack some of these different applications throughout the course and talk about how stem cells really are changing the face of medicine. Now, I think one really, really important thing to consider again here in the introductory lecture is that not all stem cells are created equal, right? And the reason I say this is because if we go back to our original definition with those 2 rules of what it takes to be a stem cell, the first is that they're self-replicating, right? And this is true for all stem cells. But the second part of that definition is their ability to give rise to other cell types. And this is where not all stem cells are equal to others. So there's stem cells that can differentiate into almost everything, right? And then there's stem cells that are more restricted that can only give rise to certain cell types. And so to help define this, we use certain nomenclature, right? Which you may have seen before, whereby if I say a stem cell is totipotent, it means it can give rise to just about everything, right? Not just the embryo, but all those extra embryonic tissues as well, like the placenta and the amniotic sac. Kind of one notch down from that all-encompassing totipotent capability are stem cells that are pluripotent, right? And pluripotent stem cells can give rise to most cell types in the body, right? In other words, those that derive again from endoderm, mesoderm, and ectoderm lineages, as you see on this slide here, but not the extra embryonic tissues. And then finally, the last of the nomenclature I wanted to mention is multipotent stem cells. And these are a bit more limited, shall we say, in that they can only give rise to cells of a specific endoderm or mesoderm or ectoderm lineage, right? For example, a multipotent stem cell that's an endoderm progenitor can only give rise to endoderm lineages, but not ectoderm or mesoderm. And so the take home here really is that not all stem cells are created equally. Another nomenclature classification for stem cells is how they're derived, right? Their derivation. And, I think this is especially true nowadays, and especially important nowadays with all the modern technology that we have at our fingertips, that we categorize a few different types of stem cells. Again, we've got these embryonic or fetal stem cells at the top. And as we talked about, these are derived from fetal tissue or from embryos specifically. Here again, we go to that inner cell mass from the blastocyst. So taking those big nucleated cells at the top here, right? And then growing them in culture. Again, these cells are pluripotent in that they can differentiate into almost any cell type in the body. And they can also renew themselves indefinitely. In the middle here, we've got our iPSCs, our induced pluripotent stem cells. And we're going to spend most of our time in this course talking about these reprogrammed cells that, again, are also pluripotent and, again, can also renew themselves indefinitely. And then lastly, at the bottom here, we've got adult stem cells. And again, these are isolated from things like tissue, organs, or blood. And they're a bit more specific in that they can give rise to only related cell types, like things like hematopoietic, neuronal, or mesenchymal stem cells, for example. And these do not renew themselves indefinitely, as the other two do.
Hey,
Now,
Tom.
yeah, Omar, please.
Sorry, one more thing here. And you'll see me chiming in every once in a while, so I'm going to apologize in advance. But
No, we want you to.
yeah, one quick thing that I wanted to point out is that ESCs or embryonic stem cells and iPSCs, it's important to kind of at least be aware that those are transient populations in a developing embryo, right? These are not populations that persist on as the embryo develops. Whereas adult stem cells, you can actually find adult stem cells in our current bodies that replenish certain cell types, right? And the distinction I make here is because and we'll talk a little bit about this in future, today actually, and in future lectures, but it's important to make sure that if you're dealing with a transient population, that you keep them in a certain state by providing certain nutrients, like Tom was saying, right? So this is where the media supplementation, the routine you're putting them through, how you're passaging them becomes very important. Otherwise, that transient population might, you know, move down that differentiation pathway when you don't want it to, if you don't hold these practices solidly in place. And we'll talk a lot about this, but I just wanted to point that out, that naturally, like in our current bodies, so you don't have embryonic stem cells or pluripotent stem cells.
Thanks, Omar. All right, so we were talking a little bit about stem cell derivation, right? And I think this slide's just a nice example of how we can dissect embryonic cells from that inner cell mass and then plate them onto a dish, as you see here in this image. Again, we call these embryonic stem cells because they're derived from an embryo. And again, these cells are pluripotent because they can differentiate into almost any cell type in the body. We can also, though, obtain embryonic stem cells by removal of a single cell from the blastomere, right? In this case, the egg's fertilized by a sperm, it grows, and then it begins to divide. And then right around this 8-cell stage here, right, of the embryo, we actually separate these cells. We can take one cell out, and this is called a blastomere. We can then culture that blastomere in the lab, and this again allows us to give rise to the entire embryo, right? And so the take home here with this one is that with just one cell, you have a colony that starts to form and differentiate, ultimately giving you another stem cell line. Now, the second aspect of stem cell derivation that I wanted to very, very briefly mention here is the process of somatic cell reprogramming. Again, so in this case here, what we're doing is taking a differentiated cell, right, like a skin or a blood cell, for example, and then we force the expression of certain protein transcription factors, which allows us to reprogram those cells back in time almost into induced pluripotent stem cells. And again, we're going to talk about this in a good amount of detail in a later lecture. But this is a process that allows us to take these cells, revert them back to an induced pluripotent state, as you see at the bottom here. And then on the right, you can see these kind of nice looking round cells, right? These are often indicative of induced pluripotent stem cell colonies, which again were reprogrammed from these surrounding cells, right? These fibroblasts around the edges here. And again, we're going to talk a lot more about what these cell types might look like or should look like, perhaps. We'll share some experiences of where these cell cultures have certainly gone wrong, which I'm sure has happened to all of you in the lab. And we'll also talk about what characterizes them as stem cells from things like a morphological standpoint. And then lastly, with regards to stem cell derivation, right, we have our third cell type that we've been talking about, the adult stem cells. And again, these are those multipotent cells that are isolated from, say, tissue organs or blood. You can see that within different parts of the body, these adult stem cells reside and actually keep proliferating, not always, but oftentimes to help replenish the population. One example of this would be in the brain, right, where you've got neuronal progenitors in certain areas of the brain that act as stem cells. And most of these stem cells are found in tiny little niches where they develop. So under the right circumstances, right, things like signaling factors, cell cycle proteins, even environmental stimuli, you can actually jumpstart these different stem cell niches in areas of the body like the brain. What's really, really cool about that, I think, especially in these neural stem cell niches, for instance, is that they can give rise to quite a few different lineages here. So different subsets of things like neurons and glia, so oligodendrocytes and astrocytes, for example. And again, this was kind of the focus of a lot of the work that I did for about a decade or so in academia. I looked at different factors that can help us remedy brain injury in premature infants, right? So babies that are born early, and how we actually tap into different stem cell niches in the brain, and then apply a lot of these principles actually in the neonatal clinic in patients, and oftentimes in some of the work that I did, quite remarkably, get those kiddos back on track from a neurodevelopmental perspective. So stem cells are super, super cool. That's the take home here. We've introduced a couple of different types of stem cells, though, but I did want to touch on a little bit of their history, because we kind of have this really cool and storied history. And I think one question that Omar always loves to ask people is how old do we think the concept of stem cells is, right? In other words, how long have stem cells been around? Anyone have any idea?
See if anybody gets close. I don't think I've ever had anybody actually come close, but to make it a little bit more specific for you guys, when do you think the first mention of a stem cell, for example, was in the literature? Was it the 90s, the 80s, the 70s? See, we have a couple of folks. We have this 1960s, 100 years old, 1970s, the 1900s, the 60s, the 20s, the 20s. A lot of good ones, but I think there's one person that's gotten close. Tom, sorry, you can continue, Tom.
No. This is great.
I
This
just
is great.
love that question.
This is exactly what we want. Well, I'm going to burst everyone's bubble. I'm sorry. The first described concept of stem cells, I think Omar told me this in a bar once, by the way, dates back to about the 1870s. There was a German scientist by the name of Ernst Haeckel. Haeckel? How do you say it, Omar?
Ernest Haeckel, yeah.
Yeah, right. He used a term called Stamzelle, Stamzelle, right, to describe a fertilized egg as the origin of all other cell types within an organism, which is pretty cool. The 1870s, that's when we kind of got this first idea. Obviously, there's been a lot of work done since then, but the next, I would say, big leap in the field was in 1953. 1953 is also coincidentally the same year that Watson and Crick discovered DNA's double helix. But it was also the same year that two Canadian researchers, and Omar's Canadian, so I'll give him a shout out again here, two folks by the name of LeBlond and Claremont published a really, really seminal paper that basically helped us identify the theory of stem cell renewal. They discovered and described a population of cells responsible for the continuous production of sperm in rodents. So stem cells have been around for ages, right? We've made loads of leaps and bounds over the course of stem cell history. And I'd say sometimes to really, really capture the public eye, right? And one good example of that was the cloning of Dolly the sheep. This occurred in 1996. Dolly was part of a series of experiments at the Rosalind Institute in Edinburgh, Scotland. And in this particular instance, researchers were looking at different techniques to help produce genetically modified livestock, right? And so what they did is they took a nucleus from one cell, they put it into the cytoplasm of another, and then they put that clone cell into a surrogate mother. They then evaluated whether the baby resembled the original sheep that they took the cells from or the surrogate, right? You can see here that Dolly has a white face, right? Whereas you can see over on the left here, which was different to the surrogate mother, the baby had a darker coat, right? Demonstrating then that Dolly was not the offspring of the surrogate, but of the donor cell that they had used. Another good example was in 1998. So scientists discovered that they could take cells from an embryo and they could then grow them in vitro, right, in a lab setting. And perhaps I would say the biggest discovery, and maybe one of the most important discoveries in science and medicine of quite a long time now, was in 2006 when Shinya Yamanaka and Jamie Thompson, I should add, really catapulted stem cell research by introducing an entirely new way to reprogram cells all the way back to a pluripotent state, right? It's quite an incredible concept, and we'll talk about this quite often throughout the course, right? And this is some pioneering work, stem cell reprogramming, right, and iPSCs that rightfully so, I would say, garnered Yamanaka a Nobel Prize, which he won in 2012. Kind of touching on that a little bit, right? In today's, let's call it a world of modern medicine, stem cells hold an awful lot of potential, as we've alluded to earlier, right? And this especially holds true in areas like regenerative medicine, drug discovery and development, developmental biology. But the recurring theme here really is recapitulating the human state through relevant models, and then harnessing that power of stem cells to help us understand and treat human disorders, again, all in a physiologically relevant manner. And to put a price tag on some of those ideas here, right, and some of that potential, the global market for stem cells at the moment is around $15 billion, which is a lot of money. And you can see, as we put on here, it's expected to grow quite rapidly over the next 10 years or so. And so really the take home here is stem cell research is a really exciting and rapidly expanding field to be involved with. So kudos to you guys. Along those lines, I'm going to segue a little bit here, and we'll dive a little bit deeper into the pluripotent stem cell workflow. And we're going to take a little bit of a deep dive here on some of these pillars or buckets of this stem cell and disease modeling workflow. And we'll do so in more detail as the course progresses over the next month or two. But what I want to do today is just briefly dip our toes into each one of these buckets here in red, as you see, just to test the water a little bit. And I want to tell and keep this a comprehensive story if we can, to really help put things into context as the course progresses. And diving into that workflow, this is a slide that I always like to start a lot of talks with, because I think it's really important, first and foremost, to call out that really underlying the utility of any viable cell model that we're using is the capacity of that cell model to predict outputs that ultimately translate to organisms, right? In other words, asking the important question, is the model that you're working with physiologically relevant, right? This is hard to do. We know that humans are kind of this finely tuned orchestra, if you will, of about 37 trillion cells, right? We also know that the genetic code, for instance, helps substantiate the basic structure and function of the body, right? But understanding and ultimately connecting how all these different inputs align with all these different outputs, right, is really difficult, let alone the fact that we can't ethically or practically experiment on human beings outside of, say, the strict guidelines of clinical trials. But there are nowadays a lot of outputs that we can measure. Put a couple of them on the slide here. Things like symptoms, biomarkers, morbidity, mortality outcomes, and then importantly, models that we generate based on those different outputs that then allow us to sort of reciprocally decipher things like gene expression, protein function, the impact of media systems, cell phenotypes, right? And so the take home here is that this is very much a reciprocal system. And as selfish as it might sound, we strive to relate all of these results back to the human being in the middle here. So one of the keys to successful culture is creating the most realistic environment possible, right? But we have to do this in a way that the cells can still be analyzed. And so, as I've kind of jokingly put at the top here, in the grand scheme of things, we've got at our disposal quite a number of different model systems, and I've only put a couple on here, to study human disorders. Starting on the left here, we've got planar or 2D culture models. These are readily available, they're easy to manipulate, but 2D culture models often lack a lot of the cellular architecture and diversity that's, I would say, necessary to mimic human disorders quite closely. And this is especially true in areas of the body like the brain. We also often employ behavioral studies, right? Nowadays, these are often performed in rodents. They're a nice bridge between, say, the inner workings of a particular organ, as well as functional output, but they're by no means directly translational, right? That's important because at the end of the day, we're still dealing with animals, right? From a neuro perspective, for instance, too, keep in mind that a lot of, say, adult onset disorders, think about things like neurodegenerative disorders like Parkinson's disease, which we'll talk about today, Parkinson's impacts our physiology long before behavioral changes are first detected, right? We also have human tissue, which is great, right? We're always looking for this as a way to study the human disorder, right? And the human systems, but it gives us a nice intact structure and therefore 3D microenvironment. But of course, it lacks the functionality component because we're typically not performing experiments in live tissue, right? There's also limited availability then with the amount of things like post-mortem tissue that researchers can actually get their hands on. We also often employ 3D models, right? These are 3D cell cultures that help us replicate in vivo conditions. I would say much, much more accurately than 2D systems. And cells cultured in 3D typically give us data then that are more physiologically relevant, right? In other words, closer to that of cells in our natural environment. We'll unpack some of this again throughout the course, but within the stem cell space, there's really two types of 3D models that we work with at Thermo Fisher, and those are suspension culture and organoids. Now, we kind of touched on this a little bit as well, but getting cells for in vitro modeling has historically not been the easiest thing in the world to do. But with the advent of iPSC, or induced pluripotent stem cell technology, there's been some really, really amazing advances in now creating models that correlate well to human disease. And so on the left here is this notion that you can take a healthy donor. From this donor, you can take blood or skin cells, for instance, and then, as I alluded to, through the forced expression of certain protein transcription factors, actually reprogram those cells back into iPSCs, right? We can then edit those cells to introduce a mutation or even revert a mutation. And then, I would say quite remarkably, hone in on the contributions of even a single gene with respect to a particular disease. And again, towards the end of this talk, I've chosen an example of that in the context of Parkinson's disease. Now, as you see across here, right on the top right, skin and blood are two common sources for iPSC generation. And then I've listed a few important considerations here in actually attaining those human cells for in vitro modeling. Things like patient comfort, the sample size or cell number desired, upfront manipulation requirements, the desired reprogramming method. I'll show you a couple of those in a moment. And of course, importantly, the intended downstream application. So once we have cells for in vitro modeling, the next kind of bucket, if you will, in our workflow is reprogramming, right? And again, that's that process of reverting a somatic cell or adult cell back to a pluripotent state such that it now behaves like a stem cell, right? And again, this became famous in 2006 with Shinya Yamanaka and co., who were able to turn somatic cells into iPSCs by forcing the expression of 4 different transcription factors, right? OX4, SOX2, KLF4, and c-MYC. Again, he won a Nobel Prize for this work in 2012. Now, you can see here on the left, there's loads of different ways to deliver these factors into cells to start that process of reprogramming. And so, as a researcher, how on earth do you pick which of these to use? Well, I put a few important considerations over here on the right. I'd say first and foremost, the safety of the method, right? You've also got to think about the consistency of the system, right? The workload of the reprogramming method, the cell type that you're reprogramming, right? There's lots of different considerations. And obviously, we're not going to attempt to cover everything on this slide, and we will go a bit deeper on some of these. But from a bird's eye view, I really wanted to take home here that you can deliver factors here through 2 main ways, integrative and non-integrative methods. As you see in this red box here in the top in the middle, I'd say more traditional technologies, things like lentiviral delivery, what those do is they integrate into the genome of target cells. What that means is that the resulting iPSCs and then cells differentiated from those iPSCs will then contain foreign DNA, which is fine, right, in some instances, but it can be problematic for use in things like cell therapy and drug discovery applications. We've also, thankfully nowadays, got these non-integrating methods, as you see in these green boxes along the edges here, that allow us to generate iPSCs that do not contain detectable vectors or transgenes, right? And the most efficient of these nowadays are things called virus particles and episomal DNA vectors, which I've circled here in red. And both of these are systems that we employ at Thermo Fisher and allow us to support the reprogramming of a wide range of somatic cells. Now, again, moving along our stem cell workflow here, the next bucket we're going to dip our toes into is genome editing of iPSCs. Genome editing, I think, is one of the coolest areas of stem cell medicine, and I think one of the areas where there's a lot of uncharted territory. There's going to be a lot of changes, and a lot of really, really cool things are going to come from genome editing in the next, I'd say, decade or so, because I think we're just at the tip of the iceberg in understanding genome editing. But at the end of the day, it's really a take-home here is that it's an approach in which the genomic sequence, right, as we see here, is changed, right? We change these sequences by adding, replacing, or even removing these DNA bases, right? But you have to consider at the end of the day that, as you might imagine, the genome is quite resistant to change, as it should be, right? Because in the body, DNA, as we've talked about, is responsible not only for encoding all the necessary functions within a cell, but it's also crucial to determining differences between individuals. And so if DNA was really, really easily altered, right, a lot of different cell functions could be disrupted, often in undesirable ways. And so to protect against, I'd say, inadvertent changes, what cells have done is developed inherent mechanisms to help proofread and repair their genetic code. And this is what we tap into when we actually perform genome editing. When we use genome editing to introduce different desired phenotypes, for example, into different cells and organisms, what we're able to do is create even better cellular models within a wide array of applications, some of which I've listed here. Think things like functional genomics, epigenetics, drug discovery, cancer research, right? Just to name a few. Again, we're not going to go into too much detail here, but because we're going to talk about this down the road, but I did want to touch on genome editing with regards to two different things here, designer engineered nucleases, right? And these include strategies like CRISPR or TALENs, which I'm sure some of you have heard of, that can be designed to target nucleases to specific sites in the genome. And then we use those to create double-stranded breaks at desired locations, as you see in the top here in this red arrow. Following these double-stranded breaks, what happens, as I alluded to, is the natural repair mechanisms of the cell actually start to heal the break. And this is typically done by one of two mechanisms, as we see here. And so big picture here, genome editing is routinely achieved nowadays through the use of things like CRISPR technology and TALENs. Again, both of which program target nucleases to specific sites in the genome and then allow us to create double-stranded breaks at desired locations. We'll touch briefly here on CRISPR, right? And I'd say CRISPR is probably the most popular and arguably the most famous of these designer engineered nucleases. CRISPR stands for clustered, regularly interspaced palindromic repeats. Absolutely no need to remember that whatsoever. Just remember it as CRISPR. But this is an RNA-guided DNA nuclease system. It was actually originally identified as part of a bacterial immune response pathway. Right, they're part of the bacteria's adaptive immune system that helps protect them, that bacterial organism, against invading DNA. What scientists did, though, is they gained an understanding of that molecular pathway, and then they kind of pinched it or co-opted it for use in eukaryotic engineering, right? Very clever. What we need for this CRISPR system is first an RNA-guided DNA nuclease system, so bacterial enzymes that cleave that double-stranded DNA. The nuclease or enzymatic activity conducted by a protein. This protein's called Cas9, right? You can think of Cas9 as a pair of molecular scissors, if you will, right? That cuts the two strands of DNA at a specific location in the genome, and therefore enables bits of DNA to be added or removed. We also have a piece of RNA called a guide RNA, which is designed to find and bind to a specific sequence in the DNA. The guide RNA has RNA bases, as you might imagine, that are complementary to those of the target sequence, and that means, at least in theory anyway, that the guide RNA will only bind to the target sequence and no other regions of the genome. And so this guide RNA or gRNA consists of a small piece of a pre-designed RNA, about 20 bases long or so. It guides or recruits that Cas9, those molecular scissors, to the right part of the genome based on a pre-designed sequence, making sure that the Cas9 enzyme cuts across both strands of that DNA at exactly the right spot. So the take home here for CRISPR-Cas9 is it's a novel gene editing system. The system consists of two parts, Cas9 enzyme and a guide RNA, which allow us to modify, edit, delete, or even correct specific areas of our DNA, again, by capitalizing on an endogenous repair mechanism. The other gene editing system here on the right that I want to very quickly mention is called TALENs, right? TALENs short for transcription activator-like effector nucleases. Again, absolutely no need to remember that, but these are based on effector proteins secreted by, again, certain types of bacteria that rewires host transcription and are actually known to cause major diseases in crops and plants, right? So these TAL nucleases or proteins contain different amino acid repeating motifs with variable positions, which is cool in this instance because this allows us to recognize specific DNA nucleotide sequences essentially allowing for unrestricted targeting of any sequence in the genome. And a big part of why these talons can be addressed anywhere in the genome is because they're a bit more general. They don't have the same constraints that CRISPR systems do. So talons, I would say, are probably limited to more simple mutations, unlike CRISPR, which can introduce multiple gene mutations concurrently with a single injection. CRISPR has a slightly higher efficiency, whereas talon editing often results in what's called a mosaicism, where we might see a mutant allele present in only some of the cells that are transfected. All right, one other thing to talk about here that's quite important, I would say, to always think about when you're performing stem cell culture is characterization of your stem cells. Characterization is something that should be done at certain intervals throughout the stem cell workflow, often depending on the complexity and the scale at which you're performing stem cell research. But at the end of the day, it always involves a variety of methods to help us identify and analyze different properties of stem cells. Think about things like identity, pluripotency, functionality, for example, right? So characterization of cells is critical as you move through the stem cell workflow. And we typically recommend performing evaluation of factors like genomic analysis, pluripotency, and sterility at these major points, right? Especially in a potential clinical setting, as you see here in the cell therapy workflow. What that does is help us ensure things like safety and efficacy, right? By confirming things like cell identity and functionality, because we can then assess things like the health of our stem cell cultures and then detect any changes or contamination that might affect their properties. Also, of course, determine their suitability for use. It also allows researchers to help understand how stem cells are going to behave, right? And this includes different capabilities like their ability to self-renew, their ability to differentiate, their ability to respond to different signals, right? And so basically these allow us to ensure these stem cell characterization techniques that our results are reproducible and comparable across different laboratories and studies, right? Which of course is super important.
And
Yeah, please, Omar, go.
sorry, Tom, I just want to highlight just one quick thing as well here. This is specifically important when if you remember, I mentioned that iPSCs or PSCs in general are transient population, right? Um, so identifying and characterizing and placing them on that spectrum, if you will, is really, really important because, as Tom said, these cells tend to move through the pluripotency potential or differentiation bias potential across development. So making sure that they're pluripotent will really help you in your downstream applications, making sure that you are able to give rise to the cell types that you want. Instead of having your cells be restricted to only a subset of populations that they can give rise to, like a multipotent stem cell would. On a PSC side, we're really trying to keep those cells as pluripotent, making sure that they are able to give rise to whatever cell type in our body that we are going to be working towards. In addition to the pluripotency state, the other big thing to consider is karyotype, right? It is very, very important, specifically also referencing what Tom mentioned about creating a very physiologically relevant model in the lab. So if we end up with a cell line or a PSC cell line that has a bunch of mutations, right? We may not necessarily be able to draw a definitive conclusion from our data because we wouldn't know whether the outcome of that particular data that we got is a result of the mutation that they had acquired or as part of the process that we're trying to study and understand, right? So just keep that in mind that stem cell characterization, really knowing the fact that your stem cells are pluripotent and are karyotypically normal is really, really, really important and is actually being emphasized across the board by the International Society for Study of for Stem Cell Research, as well as a lot of the big publication journals are asking for that characterization data to be part of the submission process with manuscripts. So please keep that in mind.
Thanks, Omar.
Of course.
And then lastly here on the topic of characterization, just to kind of put some of this into context, I put this slide in here, which admittedly has far too many words on it. I hate slides like this, but it kind of is a nice overview, I would say. that allows us to emphasize the importance of characterizing PSC lines, right? And this is especially true after what are stressful interventions on stem cells. Think about things like reprogramming, genome editing, or clonal isolation, right? Those put a lot of pressure and a lot of stress on the cells, right? Some of the recent studies, we pulled this from the literature, have shown that about 20% of pluripotent stem cells can actually accumulate chromosomal abnormalities, especially gains in chromosomes 1, 12, 17, and 20, as well as the X chromosome, right? This can occur especially in things like culture adaptation. We also see subchromosomal aberrations or alterations, right? Usually during reprogramming and culture of PSCs is when these might occur. But, we've got quite thankfully all these tools to look at stem cell characterization nowadays. So the take home here is we want you to employ stem cell characterization at routine intervals throughout your stem cell culture. It's super, super important when you're working with stem cells, and especially considering the repercussions of not doing stem cell characterization and the impact it can have on your stem cell culture. I mean, you can literally waste, and we've all had experiences with this, I'm sure, months and months and months of your life and your stem cell culture's life, right? And a lot of money from, say, a grant or funding perspective by not taking care of your cells, by proper culture, and also by checking in on your cells by routine characterization.
I'll give you a quick anecdote. Sorry,
Yeah,
Tom.
please. Yeah, no,
Just a
don't be
quick
shy.
anecdote here, not necessarily related to abnormality specifically, but just the importance of really knowing what you're working with from a cell source, right? During my graduate work, I was working with a particular line that I thought because I was studying female reproduction and then placental development. So I needed to use female lines, right? That had that were capable, right, of reproducing that physiologically relevant model in the lab. But I was working with a particular cell line for I think 2 or 3 months before I found that this particular cell line was from a previous person in the lab that was banked, and I guess there was some mislabeling of vials, and I ended up working with this line for about 2 months before I found out it was a male line, right? That didn't really, I couldn't really use because it didn't make sense in my experimental paradigm. So, something as simple as that could really, have saved me so much time and effort. So, this is why we're calling it out as best practices to make sure that you really know what cell line that you're working with, what that karyotype looks like, what the pluripotency phenotype looks like as well, before you move on to downstream applications.
Absolutely. All right. One other thing to mention here is that, especially in modern stem cell workflows, they often extend beyond things like reprogramming and gene editing, because in, say, disease modeling, especially, right, these iPSCs are almost invariably differentiated into specific cell types, with or without those genome edits. Again, as I mentioned in the beginning, to help us recapitulate disease relevant phenotypes. And so once we've generated all our cells, right, whether that's an adequate working bank or a bank of cryopreserved cells, what we see a lot more of nowadays is folks differentiating induced pluripotent stem cells into mature functional cell types. There's a number of factors, as we see here, that govern this stem cell process of differentiation, right? Which I'd say at its core is a process that involves the changing of a cell to a more specialized cell type, and one in which we see sort of a switch, if you will, from proliferation to specialization, right? And this is driven by a combination of different factors, often genetic factors, environmental cues, right? Think things like complex signaling pathways, as you can see on this slide on the left here, right, that help regulate gene expression. There's also massive influence of the extracellular environment, right? We know that the microenvironment around a stem cell plays such a big role in directing differentiation. We've got all these different factors at play, right? These physical, these mechanical forces, extracellular matrices, membrane potential, metabolic activity, cell-to-cell context, you name it, right? And all of that really, really impacts this differentiation process. But I think this is a really nice slide here, because what that amounts to is quite remarkable. You can see this over here on the right. And again, we won't go into detail here, but this is a really nice visual representation of those endodermal, ectodermal, mesodermal germ layer lineages as they differentiate into the various cell types of the body. So at Thermo Fisher, we offer a couple of different media systems and kits that support differentiation, right? And these, as you see here, include our essential 6 media, which is a growth factor-free base for differentiation reprogramming of various cell types. And then also more directed differentiation, which we achieve using things like ready-to-use kits to, again, help steer iPSCs into cell types of different lineages, right? Definitive endoderm and cardiomyocytes, for example, as you see here, or neural stem cells, which can be further differentiated into hindbrain-like neurons and glia. And of course, importantly, in the context of our time today, midbrain floor progenitors and dopaminergic neurons, which we use to study Parkinson's disease, which is as good a segue of any, because that's what we're going to be talking about today for our case study. So Parkinson's disease, which I'm sure many of you have heard of, or perhaps even know someone that lives with the disease, is a really, really common neurodegenerative disorder that's characterized first and foremost by a host of different motor symptoms. When I say motor symptoms, right, think about things like shaking, slowed movement, which is called bradykinesia, muscle rigidity, perhaps an abnormal posture and pace. And what happens with these motor symptoms is they often begin quite gradually, right? But almost invariably, they get worse over time to sort of pile on and add different disturbances on top of those in things like behavior, emotions, sleep, and memory, for example. And why this happens at its core is because neurons in an area of the brain called the substantia nigra, right, which is an area of the brain we know is responsible for things like reward and movement, those neurons in that area basically become impaired and die. These are also the neurons that produce a substance called dopamine. Dopamine, which I'm sure many of you have heard of, plays a massive role in how we do things like we think, we plan, and we feel. To add one other layer to that here, people with Parkinson's also often lose the nerve endings that produce a substance called norepinephrine. Norepinephrine is super, super important because it helps mobilize the brain for action. In doing so, then it helps dictate how we react to things like stress and anxiety. And so when we talk about Parkinson's, right, it's essentially the loss of dopaminergic neurons and then the blunting of those nerve endings that really serves as the harbinger, if you will, that throws absolutely everything else out of whack. And if we think and talk about basic functions of the brain, right, sensation, perception, feelings, thoughts, behaviors, right, you can imagine how loss of those important dopaminergic neurons and the blunting of those nerve endings, right, could potentially become catastrophic, but also as well how different stem cell derived dopaminergic neurons might offer tremendous potential here as a treatment for this disorder. And so, again, there's loads of potential for stem cell derived dopaminergic neurons in areas like disease modeling, drug discovery, or even functional engraftment into the brain. And there are companies that are working on this at the moment in human beings and have had some success as well. From a high level, though, that stem cell derivation workflow might look like something that you'd see here across the top. And then I've included here just a handful of recent papers that highlight stem cell therapy as well as neuroplasticity in these Parkinson's-associated patient populations. There's obviously loads of anticipation surrounding, I would say, not only this type of work in Parkinson's, right, but also in the treatment of other neurodegenerative disorders that we're seeing throughout the lifespan, right, which would obviously benefit quite tremendously, I would say, from stem cell therapy. And so lastly here, what Omar and I wanted to do was put some of these principles that we've been talking about today into practice. What we have here is kind of an overview slide, if you will, of a case study that was a collaboration between the Parkinson's Institute in California and our research team at Thermo Fisher Scientific. And what we did in this specific example was looked at the ability of stem cells to differentiate into dopaminergic neurons, right, which, as we mentioned, are super important in the context of Parkinson's disease. We also highlighted the importance of something called LRRK2 here. LRRK2 is a widely expressed protein, and I would say a very, very crucial factor to understanding the cause of Parkinson's disease, because we know that people have generated a lot of data linking LRRK2 to a lot of pathogenic mechanisms underlying Parkinson's disease. So things like oxidative stress, the inflammatory response, mitochondrial dysfunction, just to name a few, right? And so really, if you think about it, one promising approach to help us understand and potentially even treat Parkinson's disease is to solve this LRRK2 problem. So what we did here in this case was we used a Cas9 expressing iPSC line, right? We then genetically engineered those cells to introduce a point mutation in this LRRK2 gene here, specifically a SNP conversion called G2019S, right? That we know is associated with Parkinson's disease. We then took those iPSCs, we characterized them as we mentioned, that's super important. We differentiated those into dopaminergic neurons, and then we performed a number of different cell-based assays to help us investigate the effects of this mutation on things like the biology and function of these subsets of neuronal cells. So after introducing the mutation, our LRRK2 mutation, what we did first was confirm gene editing or gene targeting by Sanger next-gen sequencing, as you see here in the bottom left. We then, as we talked about, ran a few different characterization assays to help us confirm that the edit didn't affect things like cell quality or influence stem cell identity. In our case, this included tests like the TACMAN scorecard assay, as we see here, to help examine trilineage differentiation potential, right? So could those cells initially still become mesoderm, endoderm, or ectoderm cells, right? You can see here that the parental mutant lines were similar, which is what we wanted, and as well as the pluritest and karyostat assays here. And collectively, these allow us to basically confirm that things like pluripotency and karyotype in this case were unaffected by genome editing, as shown here over on the right. After we made those edits, what we did is we took those control mutant lines and we differentiated them using a kit that we have, our PSC dopaminergic neuron differentiation kit. You can see that both differentiated normally here, and they showed appropriate staining for different neuronal markers, things like TH or tyrosine hydroxylase here. In the second column, TH is the rate-limiting enzyme in dopamine synthesis. It's a widely used marker for dopaminergic neurons. We also looked at beta-tubulin, specifically beta-3 tubulin here, which is found almost exclusively in neurons. We know beta-tubulin plays a really, really important role in things like structural support and transport of elements like neurotransmitters. And again, you can see that these different markers were comparable in both the top panel, the mutant line, as well as the control panels across the bottom. Now, what we did next is looked at neurites. And neurites are essentially neuronal processes, right, or projections from the cell body of a neurons. What happens is when neurons grow, they essentially tailor these different projections, right, in response to different guidance cues from both the internal and external environment. So they're super important in the functionality and behavior of these cells, right? So we looked at, in this case, analyzing neurobiology and function, neurite outgrowth, using, in this case, a live cell imaging system. What we found, as you might imagine, was actually a slower rate of neurite outgrowth in the early time points for the mutant, as shown here in this red box, right, and the top figure here on the right. But actually what happened is when neurite outgrowth actually stabilized over time in normal cells, the mutant cells actually grew longer neurites initially than both the control parental lines and another mutant line that we tested, which were associated with Parkinson's disease as an additional control. And so we show here that this particular LRRK2 mutant phenotype actually ultimately resulted in longer neurite outgrowth in these Parkinson's dopaminergic neurons. And this happens likely as a compensatory mechanism or sort of an adaptive response, if you will, to injury. One of the hallmarks, though, that I talked about initially was in Parkinson's disease, right, that loss of dopaminergic neurons. And so in order to help us further recapitulate the disease, we looked at the sensitivity of our differentiated cells to different known toxins, right? Toxins like 6-hydroxydopamine here. In this case, 6-hydroxydopamine is a neurotoxin. It's specifically taken up by dopaminergic neurons. So in this case, we used a caspase high-content imaging assay, as you see here, and asked how these control and mutant dopaminergic neurons would respond to this neurotoxin. We also incorporated something called staurosporine in the middle here, right? Staurosporine is a potent, non-selective apoptotic agent, right? Meaning that we're able to use it as a reference baseline for overall cell death in both our control and our mutant lines. We've got representative images over here on the left, and then quantified apoptosis or cell death data over here on the right. You can see over here on the right in this quantified data from the images, right, that our LRRK2 dopaminergic neurons in the second column here were much, much more sensitive to neurotoxin exposure and therefore showed increased apoptosis. We know in patients as well that stress can worsen symptoms in patients with Parkinson's disease. And so to sort of, again, mimic those stressful conditions, what we did is repeated this experiment, this time in the absence of glucose, though, as you see down here at the bottom right. And again, these LRRK2 mutants showed increased apoptosis compared to the parental line, highlighting again that Parkinson's dopaminergic neurons are much more sensitive to neurotoxins. And again, this is in agreement with the literature because it mimics a lot of what we'd expect to see in a patient with Parkinson's disease. We also know that LRRK2 mutation impacts expression of a number of different genes in dopaminergic neurons. And so in this case, we used our TechMan gene assay. And what we found is that by introducing this particular mutation, we saw an increase in expression of LRRK2 regulated genes that we know are associated with dopaminergic neuron neurodegeneration compared to the parental control line, as seen here in blue over on the left. Interestingly, though, over on the right, we saw no changes essentially in gene expression in another alpha-synuclein mutant line, which is another model of Parkinson's disease. Again, this is consistent with the literature and really, I think, highlights that changes in the expression in a lot of these genes is not only specific to the LRRK2 mutation, but also important in the induction of Parkinson's-associated phenotypes. So to summarize, really, this case study, we took our LRRK2 mutants, we generated them using our Cas9-expressing iPSC line. These edited iPSCs maintain things like pluripotency and normal karyotype after genome editing, again shown using a number of really important characterization assays. We then differentiated those cells into dopaminergic neurons, right, in which this specific LRRK2 mutation was shown to affect things like neurite outgrowth, right, neurotoxin sensitivity, as well as gene expression in those cells. But again, the take home here is I think this is a really cool study and one that has a lot of really important clinical implications. Okay, that was a lot of information, I know, in the last hour and a bit. So I'll leave you again with this, what I think is a nice overview slide. We talked a little bit about biosafety and cell culture basics. We then jumped into stem cell disease modeling workflow, where we emphasized things like leveraging stem cells to create physiologically relevant models by doing things like reprogramming somatic cells to iPSCs, genetically engineering those cells, right, through remarkable technology like CRISPR. We characterize and qualify those cells along the way, right? And then we finally differentiated those cells into functional cells, like dopaminergic neurons, that help us recapitulate many elements of human biology. And in this case as well, serve as a really nice clinically relevant model to study a disorder, in this case, Parkinson's disease. Again, thank you guys so much for joining this course. I know I speak for all the instructors here in saying that we'd be happy to take any questions or feedback that you have. I've put this slide here as a nice overview. If you want to look down at the bottom here, we've included a few useful resources down at the bottom. There is as well a wealth of materials on the Thermo Fisher website. So these are just a few that pertain to today's lecture. Again, we will be sharing this slide deck as well, but I think this is useful to have in your back pocket moving forward. So thanks everyone for your time today, and we'd be happy to take any questions.
Love all the engagement in the chat. This is awesome.
Oh, nice one. I can't see the
Uh,
chat, but
yeah, I've been trying to answer some questions
I love it.
in the time, but thank you guys for that. The other one quick thing I will mention, and you kind of you've seen Tom allude to this, and we'll touch upon it throughout the course, but the experimental paradigm that you design for your experiments is also very important. You saw in the example that Tom was giving, a lot of what we base our findings on is relevant to a particular control. So for all experiments, right, making sure that you have the proper controls. You said you saw Tom always referencing those disease model cell lines that we created by genetically engineering that LRRK2 mutant to their original parental, wild type cell line, right? So we're comparing apples to apples all the time. So we are able to draw a conclusion based on that one manipulation or one variable that we have changed being the edit that we introduce in that case, right? So just keep that top of mind as well, because it will help you throughout your scientific career when designing your experiments, making sure that you are able to really derive a solid conclusion from the work that you've built. Any other questions? It's always like if there's not a lot of questions, although there was a lot of questions in the chat, so we're off the hook there. But it's either the presentation was really good or really bad. So hopefully it was really good. I thought you did a great job, Tom. I'm a little bit biased, obviously. But as Tom mentioned before as well, please make sure that you are asking any questions that come to mind, interrupt at any time. We want this to be as engaging and as productive for you guys as possible. So please, 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 anytime something comes up, raise your hand, speak up, yell something out. You can't throw anything at us virtually, but maybe we'll see
You
if
could try.
we can get a feature on Zoom. But yeah, with that, no, Kitai or Catherine, if you have any kind of closing things for today's lecture. Oh, I see, Haley, there's some folks that are trying to go off mute, I guess now that are not able to. Can we unmute everybody or make sure that people can unmute themselves?
Yeah, I have my hand up. Can I ask a question?
Please, absolutely. Yes.
Hi, Tom. Hi, Omar.
Hi, good to see you.
Nice to see you too. You did a great job, actually, both of you. But Tom, as always, you have this really nice story and very cohesive and stuff.
Oh, thanks, Irene.
I have a simple question that might be one of the stupid ones, though. The totipotent cells, are we considering any of the ones that are cultured in vitro totipotent or no?
Typically, I would say no.
Yeah, I would say no too.
Yeah, the cultures we can get more into this, but perhaps in later lectures. But even on the pluripotent side, there's a distinction between what we call naive pluripotent stem cells or primed pluripotent stem
Right.
cells that again are across that different part of the differentiation path, if you will. So totipotent actually are still, I would say, above that. More closely to the morula stage, where there really isn't as much asymmetry breaking in that early embryo development. Even if you kind of go even at the morula stage, actually, past 8 cell embryos, the cells technically, once they have that inside out organization, they tend to also be slightly away from that totipotent stage, I would say, because you can't give rise to embryos from single blastomeres. And the reason why I say that, so totipotent cells, you saw those 4 cell embryos or 8 cell embryos that Tom was talking about, right? You can actually remove the zona pellucida, which is the casing around these cells initially in the embryo, and you can separate each individual cells. We have some really cool techniques in the lab where we use micro manipulators and lasers to actually separate those individual cells. And if you separate those 8 cells, they can give rise to 8 identical embryos again, right? Technically. And that would be what we would consider to be truly totipotent. But you can't do that in later stages, like you would in the blastocysts or with PSCs particularly.
Okay,
Great
so around
question.
that 8 cell stage, and then after that, no.
Yeah. And most folks even do it a little bit earlier, to be honest, from a 2 to 4 cell stage. Yeah.
Okay, thank you.
Definitely not a stupid question. A great question.
Yeah, there's no silly questions.
I see. Ari, you have your hand up as well. Did you have a question? Oh, I don't see it again. I don't, maybe not. There's a couple more questions in the chat. I'll do my best to answer them today as well. Let's see. We have a question about producing neural progenitor cells in the lab, best practices for MPC and organoid generation. This is from Emma. Emma, we will definitely be covering this in a later lecture, looking at ways in which you can differentiate your cells and different methods and different protocols, including 3D organoids or spheroid culture for NPCs. So stay tuned. We're definitely going to cover that pretty soon.
And Emma, if time is a critical factor and you need a couple of answers in the interim until we do the neuronal lecture, please feel free to email me, and I'd be happy to share any of those protocols or answer any questions that you have in the meantime, for sure.
Yeah. And we also have another question from Annette about whether there's a higher chance of naive versus primed PSCs being more susceptible to double-strand breaks or aberrations. To be honest, I'm not quite sure. I would say there could be a chance, but I don't know if the data that I've seen so far in the literature hasn't made the clear distinction between susceptibility of primed versus naive PSCs, specifically when it comes to DNA damage. That being said, I will say that they're in PSCs, and this is probably, in reference to that question as well. In PSCs, there are some mutational hotspots, right? We know that there are certain parts in the genome in that pluripotent state that are much more readily or that mutate much more readily or become aberrant much more readily compared to others. For example, genes on chromosome 1, chromosome 12, chromosome X and Y, we tend to see really we tend to see higher chances of detecting aberrations on those chromosomes compared to the rest of the genome. I believe chromosome 20 is one of them. And I think part of that is the way in which the chromosome is packaged, right? Or chromatin state of that particular cell type. So there could be a case as to why there would be different susceptibilities in the prime versus naive states as well. Good question.
Shall we raise the question?
What was it, Kitai? Sorry.
Oh, yeah. Shall we? Yes, raise the hand.
Oh, yes. Shelby, you had a question?
Could you unmute? Oh, she cannot
Listening to
unmute.
unmute. Let me see if I can. Maybe Haley, if you give maybe Tom and I admin privileges, we would also be able to help you out with that. But Shelby, if you also want to type in your question in the chat, I'm happy to read it and answer it for you as well.
Yeah, while Shelby is writing her question, and then anyhow, thanks for all of the participants in the Thermo Fisher and the UCLA stem cell basic training. I hope today's session has given the strong foundation in stem cell research and also its application. So as you continue to your journey in the field, and I strongly encourage you to stay curious and also apply what you have learned here to your own research and the clinical work. And then next week, we are going to talk about iPSL reprogramming and all of the other reprogramming, which is very interesting subject. So therefore, stay tuned and come back in the same time. Okay, there is a question.
So we have more questions in the chat. There's a question about whether there's any major phenotypic differences observed in stem cell cultures in their lab, especially after being cultured for a long period of time, versus newly extracted or isolated cells from an organism due to selective pressure for surviving in vitro environments that are inherently different from the in vivo environment. Arjun, that's definitely true. Right, there are definitely phenotypic differences between different cell lines across, that could be intrinsic to the cell line itself, but also to the culture process in itself as well. Whether that be, freshly cultured and isolated or generated iPSCs compared to maybe ones that have been passaged for a very long time. We're going to talk about those particular morphological differences in more detail in another lecture, but we typically look for things like how compact the colony is, how rounded their edges are, and refractive their edges are. We look at the individual cells, and we look at the nucleus to cytoplasm ratio. So PSCs tend to have, in their most optimal state, tend to have a large nucleus compared to the rest of the cell body. We also look at the number of nucleoli or the presence of nucleoli. So a lot of you will see this and might think, oh my God, my cells look weird under higher mag. There's multiple very prominent nucleoli in my cells. And that's another hallmark of actually, PSC cells. So there's definitely some telltale signs. And as those cultures kind of skew and become less optimal over time, typically, if they're kept in optimal conditions, they should look similar across the board. But as we all know, cells tend to accumulate mutations spontaneously. And so because these cells can indefinitely renew themselves, right, there is a bigger chance that throughout a prolonged culture period, that there may be some genomic abnormalities acquired. And Tom, hinted at this. There's actually some data in the literature suggesting, this was a while back, but as high as 25 to 30% of cell lines analyzed at one particular time had a P53 mutation in PSC cultures with folks that didn't even know that they had that mutation in their particular cell line. So definitely good to kind of stay on top of that characterization piece, because which we partly also do by looking at the phenotype and morphology of the cells. Absolutely. I hope that answers the question. That was a little long-winded. But there's another question about deriving cell lines from blastomeres. Given that the embryo is not destroyed when deriving the cell lines from blastomeres, why would you choose to derive cell lines from the ICM? So that process is actually very invasive. And because you have to remove the zona pellucida, if those cells aren't immediately grown and transplanted, they will eventually demise, or will cause their demise. So it's easier said than done, right? Is the moral here, for in terms of the separating the single blastomeres. Actually, when Tom was showing that slide, I had a little bit of a flashback. I had one of my mentors way back when, the way he was studying patterning of the embryo. So he would take his eyelashes, and because it was so thin, and he rigged this very simple but like old clunky way of trying to figure out how to separate those cells without laser ablation, to try to see if he can improve viability that way. And he always jokes that he only has a couple of eyelashes left because of that. But it is definitely a very tough process that may very likely end up in the demise of that embryo eventually. Okay. For studying genetic variation in disease modeling using IPCs, what would be the karyotype in this case? Great question. So in that instance, this is where having a control or a parental control is really, really important, right? Making sure that you're comparing the karyotypes of your original line versus the disease model that you made with that gene mutation. And in that way, there's multiple different ways in which you can do this. If you're looking for gross chromosomal abnormalities, like big changes, we can use things like G-banding, or there's certain arrays that some people are working on now that can be employed. But more often than not, if you need a very specific resolution down to a single nucleotide, we tend to use next generation sequencing or Sanger sequencing to confirm the sequence of that particular gene. Okay. Kita, the seems like the unmute function is disabled. I think Haley did that. That's perfect. Any other questions from the group?
And
Angel has had his hand up for a
I
while.
do. I think Thermo Fisher shut down their Karyostat service. So how do we look at changes in genome? Is there something new coming out that's better, or are you just getting out of the genome analysis game and we're outsourcing it to YCell or someone else?
Great question. Yeah, for the time being, we have paused our characterization services for the time being. It has nothing to do with the services themselves. It had more to do with an internal change on our end and some restructuring. So stay tuned. Hopefully, we'll come up with some more solutions there. But at this particular time, I think the best way would be there's the traditional G banding, which either local institutes and local cores might offer, or you can send them to places like YCell. Additionally, there are some other companies that have some decently high resolutions for their karyotyping services that they can use. I'll try to put some together and send it your way as well, or maybe to the rest of the group, just so that they have those options too.
Will there be that service coming forward in the future, or that's still up in the air?
We don't know yet, to be honest. So far, I have no idea. That's a bit above my pay grade and Tom's pay grade. So we really don't know.
You know more than
But
I do.
hopefully,
So thanks so much.
Hopefully, because they were very easy, quick services for sure.
Yeah, also UCLA Human Stem Cell and Genome Engineering Center provide the low-cost CMP analysis.
Mm-hmm.
So if you're interested, just contact us.
Excellent. Seems like we've pretty much answered all the questions for today. Any last questions? No? Okay, we're good to go. Again, thank you again, Tom. That was awesome.
No, thank you everyone for joining. This is going to be a good, fun course. So we're really happy to have you and really happy to be involved. So thanks.
Awesome. Thank you, everybody. Have a great rest of your week.
Great week. We'll see you next week.